This presentation is about the breeding of Impatiens walleriana to develop plants that are resistant to Impatiens downy mildew. Before I begin my presentation, I'd like to explain a little bit about Cornell University's campus on Long Island. The Long Island Horticultural Research and Extension Center is located on the east end of Long Island in the city of Riverhead. We are about 315 miles southeast of the main campus in Ithaca. The research center is located in Suffolk County, the leading crop agricultural county in the state of New York. There is no other county in New York that is more diverse than Suffolk County. Agriculture and horticulture industries developed in Suffolk County because of the environmental factors that are good for plant growth. There are fertile, well-drained soils. We are in a unique climatic zone of USDA Zone 7, which is considerably warmer than upstate New York. There's plenty of fresh water and underground aquifers and plenty of sunlight. With the large markets that are just to the west of Suffolk County, the agriculture community receives very high returns on their investments. The Long Island Horticultural Research and Extension Center is one of a kind in the United States. We're the only horticultural research and extension center that has professionals that represent each of the commodity areas as well as the cross disciplines. We have specialists in the greenhouse and floriculture industry, specialists in the fruit industry with a specialization in grapes and viticulture. We have specialists in vegetables as well as woody ornamentals, which covers the turf industry, the nursery industry, as well as plant maintenance. In addition, we have cross-commodity specialists. We have 2 plant pathologists for plant disease. We have a weed scientist. We have 2 entomologists for insects and bugs, and we have a plant tissue culture specialist. Impatiens walleriana was the most popular bedding plant in the United States up until about 2011. It's so popular because it comes in a variety of colors, almost every color except yellow and a true blue. It's also a plant that grows very nicely in the shade. It can even grow in the sun if it's irrigated properly. The plants are propagated by seed, which is a benefit to the commercial producers. It reduces their costs for production of this plant, and they get a higher markup. In 2010, the value of Impatiens in the United States was $151 million. In the state of New York, the value of Impatiens was $10.4 million in sales. But then Impatiens downy mildew hit the United States, and by 2011, the value of Impatiens in New York went from $10.4 million down to $1 million. Impatiens downy mildew is a fungal disease. It's from the species Plasmopara obducens. It was first reported in the United States in 2004. It entered the United States in a hurricane that traveled from Africa to Florida. The spores were released in Florida, and then the prevailing winds along the East Coast spread the disease up the entire eastern coast of the United States. By 2011, it is reported across most of the inhabited world. It's globally distributed. This particular disease can be controlled by fungicides, but the fungicides that are used are restricted in the United States and can only be applied by greenhouse professionals. Once the plants leave the greenhouse to the homeowner's yard, there is no control that works on this particular disease. This is what happens to a homeowner's planting of Impatiens. The photo on the left looks what a nice planting of Impatiens would look like around the base of a tree. The photo on the right is what they look like, the very same plants, after they've been attacked by Impatiens downy mildew. They lose their leaves, they lose their flowers, and all that remains are these stems. And about 3 weeks later, the entire plant dies. It's fairly amazing how quickly this Impatiens downy mildew will kill a plant. It's fairly easy to tell if you have Impatiens downy mildew. Turn the leaf to the underside and look. If you see a white colorations, a white fuzzy kind of colorations, that's the spores of the Impatiens downy mildew. The plants, the photo on the left in the forefront showed the two plants that are our controls in our field. Those have Impatiens downy mildew, and you can see that they've lost most of their leaves and their flowers. The plants in the background of that photo are some of our resistant hybrids. They are still green, leafy, and quite healthy looking. Once a plant gets Impatiens downy mildew, there is no cure, and it is a systemic disease. It will never leave the plant. The disease will also remain in the soil after a plant's been infected. So future years, it will re-inoculate new plants that are planted. The objective of our breeding program was to use integrative hybridization to generate resistant plants. We searched for species of Impatiens from around the world that were resistant to this disease, and then crossed them with Impatiens walleriana to produce interspecific hybrids that would be resistant. You may be familiar with the New Guinea Impatiens, which is a different species. It's Impatiens hawkeri. They are resistant to Impatiens downy mildew, but unfortunately, they are not compatible with Impatiens walleriana. In 2013, our ornamental plant pathologist, Marjorie Daughtry, did some preliminary research looking at 10 different species of Impatiens. This graph kind of shows you the results of her research. She had 5 species that were susceptible to Impatiens downy mildew and 5 species that were resistant. We followed up with this research later, a few years later, and we looked at about 40 to 50 different species of Impatiens. We collected seeds and cuttings from around the world, grew the plants, and then tested them. We set up a screening criteria to evaluate all of our plants, from 0 to 5, 0 being the best plants and 5 being the worst plants. If they were ranked with a 0 or 1, they were what we considered resistant plants. Anything above a 1, we considered to be susceptible. These are some of the plants that we evaluated. And in this particular table, you can see the column on the left are the species of Impatiens that are susceptible to downy mildew. The species on the right are resistant. We used those species in our hybridization program to cross with Impatiens walleriana. This just gives you an idea of what we did. We would take a susceptible species like the Impatiens walleriana on the left and cross it with one of the resistant species to produce a hybrid plant. In addition to looking for resistance to the disease, we also evaluated the plants on their flower color, their growth habit, and their ease of growth and propagation. This is another example shown on the top left, a hybrid of Impatiens walleriana that was crossed with a resistant species. And you can see in the bottom of the slide the resulting hybrid, which is quite attractive. During our breeding, we used embryo rescue as a technique to be very helpful for 2 main reasons. One is that Impatiens have explosive seed pods. That means as the ovary ripens, the seed will explode and scatter the seeds everywhere. By placing these seeds into tissue culture, we were able to recover all the seeds that were generated. In addition, by producing seeds in vitro, we were able to have a sterile germplasm bank for all of our hybrids for future use. The process is pretty straightforward. We would remove ovaries about 13 days after pollination. We would take them to our tissue culture laboratory, and working under the laminar flow hood, we would sterilize them in 95% ethanol and flame them. The ovaries would then be cut open, and the ovules would be aseptically removed and placed on a quarter-strength Murashige Skoog medium with only a quarter-strength sugar. They would then be labeled, wrapped with parafilm, and placed in the dark until they germinate, which is anywhere between 15 and 20 days, sometimes a little bit longer. Once the seeds germinated and we had little seedlings, we would subculture the seedlings, into individual culture tubes. We would be able to track them better by placing them in a tube and giving them an ID number. As the plants grew, we would propagate them, and then we would divide them so that we would have a clone in vitro and have a clone out in the greenhouse. The plants in the greenhouse were then grown till they could be propagated for us to sufficiently produce enough replications for the field. At about 2016, we had 2 hybrids that were generated that would be completely resistant to Impatiens downy mildew. This was the primary research of James Keach, who was getting his PhD with us at the time. We then used these 2 original plants to generate everything else that followed. We took these 2 lines, we self-pollinated them, and we cross-pollinated them. And we generated hundreds of hybrids from these plants. These new hybrids then were propagated. They were clonally divided so that we could trial them out in the field and test them for resistance. And after that, we selected the plants that were resistant. Approximately 1/3 of our plants were resistant from these first crosses. The others were susceptible, and they were tossed. Following years, 2018, we continued to self and cross-pollinate the resistant plants, grew them up in the greenhouse, propagated them, and then trialed them in the field. We discarded anything that was susceptible, and we kept the plants that were resistant. By 2019, we had 30 superior Impatiens plants that were resistant to the downy mildew. We have all colors of Impatiens except for a true white. This gives you an example of what some of our score sheets look like. We had the number of the accession number of the plant on the left column. We kept track of what the parentage was of that plant and then their average score. Remember that the lower the number, the better the plant. And anything under the number 1 was considered acceptable. Anything higher than 1 was not acceptable. This shows you how we do some of our trials. We have 2 hoop houses out in the field that are covered by 50% shade. We grow the plants in black plastic with drip tubing underneath the black plastic. They are grown outside until the downy mildew hits. Usually it's at the end of August or early September. And then we go and evaluate each plant once a week until the plant is dead. For Long Island, we often don't get a killing frost until the end of October. So we'll be out there taking data for an extensive period of time. So we're at the point now that we're taking these main plants and trying to inbreed them to produce homozygous lines so that eventually we can produce F1 hybrids. Our goal is to produce lines of Impatiens that can be propagated by seeds. This is our team from this past year, that have been working on this project. We've been very fortunate to have some great students, graduate and undergraduate students, work on this project. I'll be happy to answer questions later. Thank you.